PCR. So, in general, and I'll talk about it again.
Definitely overcomplicated that.
To make these mutations, hopefully you will get some sort of output. So what you guys are going to do today is basically try to figure out, well, start the experiment that will let you figure out whether you made your mutation successfully. So there are a couple ways to do this. The first way is just sequencing your PCR reaction. So we could send this off to a company, they could send back the sequence of it, but that's expensive and it's kind of boring because we actually have something that we can see whether you guys made those mutations or not. If you made them, hypothetically, you should see a different color. But that means that we need something that's going to actually turn a color. So in your tubes last time, you put some DNA in there, you added a bunch of other clear liquids. Do you think you'll be able to tell by looking at your tube whether your mutation was made? No.
Gotta make the protein.
There we go. Before you use the black light, you have to make the protein. DNA has no color at all. You could have been absolutely wildly successful in your PCR, and that is going to be clear liquid. Still clear liquid. So you need something that's going to make that protein. And the easiest thing to make that protein is a bacteria. So what we're going to do today is take, maybe, apparently it doesn't want to work. Except, oh, nothing wants to work. I killed it. Fail.
That's just how it works.
It broke.
Oh, all right, maybe that works. Okay, so what you guys are going to do today is take your DNA that originally had GFP as a sequence. Hopefully you've changed that. And we're going to put that into a bacteria. That bacteria is going to transcribe the DNA into mRNA. It's going to translate that mRNA into a protein. And hopefully by, what's going to be next week that you guys are going to look at these, we're going to let these grow up overnight, and you guys will be able to see bacteria that are hopefully expressing your mutated color. So it's going to look something like this, like I showed you guys last time. You guys are going to have individual colonies of bacteria that will maybe be a different color. The way we do this, these bacteria normally don't have your GFP. They're normally not green, right? Bacteria don't glow green. What we're going to do today is called transformation.
Can I pause you right here?
Yep.
If you have your engineering notebooks out and you don't have notes on transformation, you should add that. Everything that we're learning and reviewing today is all stuff that's eligible for the test, which is happening next week. So just keep that in mind next week sometime. Probably Wednesday. Tuesday or Wednesday.
Tuesday's a work day, don't make that.
Oh, we'll do it on Tuesday.
All right, so if you have your notebooks out, if you need them out, this is the general gist of transformation. We start with a bacteria that doesn't have your plasmid in it. We take your plasmid that either has GFP, so this is your piece of DNA, has GFP or your mutated GFP, along with something else that we're going to talk about in a little bit. These bacteria are special in that when we heat shock them, so we heat them up to about, this is 42 degrees Celsius, it's about 80, 85 degrees Fahrenheit. So we'll heat it up for a very short period of time, about 35 seconds. It makes these bacteria, it makes the membrane that surrounds them kind of holy, it makes them porous. So when that happens, your DNA, which is a plasmid, can sneak in through there, and then we'll put them back on ice, and they'll kind of close back up again, and they'll basically trap your plasmid inside. Be aware, this is not to scale. Plasmids are not very big. They are not half the size of your bacteria. This is just a representation. Plasmids are very, very small. So it's not like you're opening up half the cell to get a plasmid in. This is what you guys are going to do today. And we're going to talk about how that actually works and how you know whether it works right now. But before we do that, I'm going to correct some lies that I told you last time, because I lied to you a lot last time. First thing, just so you guys remember, we made mutations with PCR, right? You had your primers. One started copying this way, one started copying that way, and the mutation was right here in between them. And I told you that at the end of it, you're going to have lots and lots and lots of circles of your mutated DNA. That circle part, that's a lie. All you guys really did was make lots and lots and lots of copies of a straight line. Your primers go in different directions. They don't overlap. They were right next to each other, which means that one copy is coming around this way and it stops right here. One copy is coming around this way and it stops right here. There is nothing putting those guys actually back together. So you have a long line. It can still be in the shape of a circle, but it's not actually attached. If we don't put circles of DNA into bacteria, they get degraded. So everything that you put into a bacteria, if you want to express it into a protein, it needs to be a circle. So the first lie I told you was that we made circles. We didn't. But I fixed that by using a quick little enzyme called ligase. Oh, it's up there. Ligase is an enzyme that sticks DNA pieces together. It's very easy. I decided not to make you waste your time doing it. I added it to your PCR reactions. Instead of having straight lines of DNA, you now have circles of DNA. Ta-da! You're so welcome. So you should now have plasmids with your mutation in there. This is not your plasmid, but you get the idea. I lied to you a second time, and somebody called me out on it last week. They said, aren't we still going to have the original template in the reaction? You guys set up a PCR reaction. You have to copy off of something, right? You had to start with a piece of DNA. The DNA you started with was a plasmid that has GFP on it. It doesn't go anywhere. Just because you guys did a PCR reaction doesn't mean that it got degraded or it disappeared. It's still in the mix, which means that if we're going to transform your new plasmids into cells, is there anything that's going to stop that old plasmid from also being transformed into cells? No. And if you're trying to tell if there's a difference in color, you don't want to have a lot of green things mixed in with a lot of blue things or a lot of yellow things. We want to get rid of all this old stuff. We only want to have your mutated plasmid. Again, there's an easy way to do this. This is called DPN1 digest. You guys use restriction digests based in your taster lab, right? You guys, if you had it, it cut at a certain sequence. This is a little bit different. It doesn't cut at a specific sequence. It cuts at a specific chemical group. So any bacteria, or I'm sorry, any DNA that is inside an organism, inside you, inside me, inside a fruit fly, it's going to have what we call a methyl group, or these little M's. This is just a carbon with 3 hydrogens on it. And it gets stuck to the outside of your DNA. So you've got your DNA strands that are connected by hydrogen bonds here. On the outside of it, you'll get methyl groups. This is just natural process. It's very important for biological functions. If you don't have methyl groups, something is going very, very, very wrong. But it gives us a way to differentiate between your original plasmid, which was made in a bacteria, and your new plasmid, which was made by PCR. Things in a test tube don't have methyl groups. Things in a test tube are just pure DNA. So we can use a restriction digest enzyme. This one is called DPN1. And again, added this to your PCR reactions. And what it does is it cuts up anything that has a methyl group. So now, remember, you need a circle to go into a bacteria and actually express a protein. These guys aren't circles anymore. Now they're just cut up pieces of DNA. So the only thing that should be in your PCR tube right now is your mutated plasmid. This is biology, nothing's 100%, so it probably won't be. But that's what it should be right now, is just the mutants that you made, none of your original stuff, and it should all be in a circle. Questions there? Are we good? Yeah.
Wait, so what comes out doesn't have the methyl group?
Yep. So.
And then we're using things, the restriction enzymes that can detect the methyl groups to eliminate those ones.
Yes.
Okay.
Yeah. So when you do things in a test tube, we don't have all the same things that we do in a cell when we're copying DNA. So when you copy DNA inside of your body, these methyl groups get added. When you copy it inside of a bacteria, methyl groups get added. The original DNA that I gave you has methyl groups because it got made in a bacteria. And then I took it out of the bacteria. The stuff that you made, all those copies happen in a test tube. No methylation, so we can now cut up the old stuff with this enzyme that recognizes those groups. Keep your good stuff.
So methylation only occurs in organic compounds, though?
It only occurs in living organisms. So you can make it happen in a test tube if you add the right stuff. We didn't.
Okay, cool.
And it won't necessarily happen in the right spot if you do it in a test tube. So yes, only in living organisms. You did not make the PCR reaction in a living organism. Okay, good there?
Yep.
Perfect. All right, so now on to today. Now that you have just your mutated plasmid, hopefully, what do you do with them? We put them inside bacteria. So bacteria have their chromosome, which has most of their genes, and then they have their own natural plasmids. And this is important because we are going to take advantage of what bacteria do all the time. Bacteria, when they divide into two cells, they copy their chromosome and they copy their plasmids, and then they split them evenly between their two daughter cells. What we're going to do is take advantage of that and put our plasmid in there, and just like a natural plasmid, it's going to get copied and it's going to divide into a new cell. And that's important because we want to be able to see it. In order to see bacteria, we need lots of bacteria. These little colonies, these are not individual bacteria. These are billions of bacteria. So each colony has to start from a single one that divided a lot. So we need these plasmids to be able to be copied and passed down. So that's important for us. So now we've got our plasmids in, we can put them in the bacteria, mwah, magic. The problem is that this is wildly inefficient. We always talk about transformation as it's an easy thing. You take your plasmid, you put it in the bacteria, you heat shock it, plasmid goes in. In reality, this happens about 1 in every 10,000 cells. Is that good? No, that's very bad. Which means that you're going to have a lot of cells that don't have plasmid. Now we're starting with billions of cells. So you have a lot of 10,000 cell chances to get a plasmid in there. But it means that you have even more cells that don't have them. And I mean a lot of cells that don't have your plasmid. That's going to give you a lot of background, and it can actually overgrow anything that you wanted to look at in the first place. So we need a way to separate out these bacteria that didn't get a plasmid from the ones that did. And this is important because we are going to start working with antibiotics and antibiotic-resistant genes. How many people have heard of antibiotic resistance as a problem in medicine? Right? You go to the ER, you get an infection, and you can't kill with an antibiotic. This is a huge problem. It's becoming a huger problem because of kind of sloppiness with antibiotic resistance in general. Now, it's a really important tool for us in the lab, but we have to be careful with it. So what you guys are going to be doing today, you have to be careful with it. You are working with living cells. You are working with actual antibiotic resistance genes. The gene that you are going to be working with makes bacteria resistant to ampicillin. Some people have probably, in this room have probably had ampicillin as an antibiotic. This is very, very, very important that you are not sloppy, that you're not spreading your bacteria around, that when you are using your tips, that they're going directly into the waste containers that are going to be on your table, that they don't accidentally get shot off at somebody or on the table. If they do, clean it up. You don't want to have this bacteria that may or may not be antibiotic resistant floating around in the lab. And it's not just for you guys. When I was in here earlier, there were a couple kids in the, not this class, but in some other class that were just like playing around with the pipette tips that have been sitting over there in the bleach. Yes, exactly. So it's not just you guys you have to worry about, it is your classmates who may or may not make good decisions in that lab space. So when you are doing this, it is imperative that everything that you touch with bacteria, everything that you touch with your plasmid goes into that bleach container right away, and then doesn't get stuck on the table or stuck on your friend or stuck on your shirt or anything like that.
Stays there.
Yes. So that's my little spouting of bacteria antibiotic resistance. Be careful when you're doing things today. You are working with living things. We're going to try to make them grow, but want them growing where we want them, not where other things are. Okay, so how do we get rid of these bacteria? I just said we did something with antibiotics, and we do that with this plasmid. So this is not your plasmid, this has a different gene on it, but you can imagine that this says GFP or BFP or YFP. This plasmid, anytime we put a plasmid in, we add an antibiotic resistance gene somewhere on that plasmid. So this one, the one that I'm giving you, is ampicillin resistant. So if a bacteria gets this gene, it can now survive when ampicillin is present.